Okay, so I'll be talking today about a critical molecule, serotonin, and I want to talk about it for its multiple uses in the body. So serotonin, it turns out, is absolutely fundamental to the growth, maintenance, and function of the bowel, and it serves as a conduit between the bowel and the brain. So Ecclesiastes points out biblically, to everything there is a season and a time and a purpose under heaven. Time to be born and a time to die, a time to plant and a time to pluck up that which is planted, a time to kill and a time to heal, a time to break down and a time to build up. So in the bowel, serotonin is a neurotransmitter. It takes care of motility, mucosal growth as well. It's a paracrine messenger released from epithelial cells in the lining of the gut to initiate peristaltic reflexes, secretion, and inflammation. It's also an endocrine hormone. It impedes osteogenesis and is involved in glucose regulation, acting on the pancreas. It's a powerful growth factor involved in neurogenesis and neural differentiation, and it's a neuroprotective agent, enhancing the survival of enteric neurons. So very much in parallel with Ecclesiastes, there are roles for serotonin that correspond to each of these. So the bowel contains an extremely large nervous system, which I have called the second brain in my book with that name, because it's the only part of the peripheral nervous system that can function in the absence of input from brain or spinal cord. Now, the nervous system of the gut is entirely derived from neural crest cells that colonize the fetal bowel. So they move from the neural crest to the gut. And they come from three regions: the vagal, which is important for the esophagus, S1-2, somites 3 to 5, stomach to the hindgut, and somites 6 to 7, the hindgut itself. Another component comes from the sacral crest, and that does only the postumbilical bowel. So these two populations of emigres to the gut overlap in the hindgut. And in addition, postnatally, Schwann cells from the extrinsic nerves move into the gut and colonize the colorectum and actually form neurons. So as neural crest cells migrate to the gut, the population that migrates, which I've labeled here ENCDC, enteric neural crest-derived cells, changes as they migrate because they're subject to microenvironmental influences that affect them as they migrate to the bowel. Within the gut, they're subject to signaling factors such as ligands and receptors that drive proliferation, differentiation, migration, and assembly of circuits. And they express a number of transcription factors that are critical in their maintenance, in neurogenesis, and specification of the cells as neurons. So the reason I call them neural crest-derived cells is that the population that comes into the gut is not at all like the population that left the neural crest. Much like the population of the United States is largely derived from emigres, which came from somewhere else. Those emigres had multiple influences on them, and the populations that reside in the lower east colon or in the United States is quite different from the original population that left the original lands. Now, serotonin-containing neurons, serotonergic and cholinergic neurons, are among the earliest born neurons in the developing enteric nervous system. And that was shown back in the 1990 using tritiated thymidine. So injections of tritiated thymidine were given during gestation, and radioautographic labeling was assessed, and you can see identified neurons. So here's one immunocytochemically shown as serotonin, and it was labeled, and it was injected on E10. This is an NPY-containing neuron injected with thymidine at E12, and you see it's not labeled. These are chat or cholinergic neurons injected at E12, and they are labeled with thymidine. So when a neuron is born, it has one final division, and that last division, the terminal division, is called the birth of the neuron. So it takes up thymidine in preparation for that last division, and after that, that thymidine, the radioactive thymidine, is diluted out if the cells continue to divide. But if it's the final division, they stay labeled for the long periods of time. So in a postnatal animal, you can see that this animal that has been labeled with tritiated thymidine at E12, embryonic day 12, is still labeled. So if you look at the birth of neurons, you see that there's a very sharp line, peak, where serotonin neurons are born, peaking even before the gut is colonized, so that some serotonin neurons are already postnatal when they arrive in the gut. Cholinergic neurons, some are born as early as serotonin, but the bulk of them are born later. After birth, some neurons continue to be born, and even this technique of tritiated thymidine birth dating can show that neurons continue to be born through the first 3 weeks of life in the mouse. That would be almost 3 years of age in a human. So the process is shown diagrammatically here. Neural crest-derived precursors in red colonize the fetal bowel. And you see these cells migrate in it. Serotonin and cholinergic neurons are born first, and you see them turning green here as a new neuron, and they put out axons. And these axons innervate still dividing cells. So acetylcholine and serotonin promote the birth, or are able to promote the birth of late-born neurons. Now, for serotonin, the gut is the big enchilada. If you look at these different organs, you see that the percent of the total serotonin of the body that's in the gut is massive. By far, the bulk of that serotonin is present in the EC, or enterochromaffin cells of the mucosa, which are epithelial cells. A smaller population is present in enteric serotonergic neurons, which are about 2 to 3% of the neurons of the enteric nervous system. About 2% of the body's serotonin is in platelets, but platelets get all of their serotonin essentially from the gut. Platelets are unable to make it, so they take up serotonin as they circulate through the bowel. The brain, where most of the research on serotonin is done, contains perhaps 3% of the body's serotonin. So quantitatively, the brain can be small, can be thought of as an small afterthought of evolution. So as I've said, serotonin in the gut is stored in EC cells. Here's an EC cell as it's demonstrated classically with silver. Here's with immunocytochemistry. And you can see that these cells have long extensions called neuropods, or feet, that extend out from them. And the bulk of the granules, which contain the serotonin in these cells, are not apical. The apical surface of the cell contains receptors that sense bacteria and other products in the lumen. But serotonin is mainly released into the connective tissue of the lamina propria, where it comes into contact with nerve fibers, which come within several micrometers of these cells. Now, 2 enzymes are responsible for making serotonin. EC cells contain only tryptophan hydroxylase 1, whereas the neurons contain tryptophan hydroxylase 2. TPH2 is a different gene, and you can see it demonstrated here in serotonergic neurons by in situ hybridization. Now, EC cells secrete serotonin. It activates reflexes in the gut, and 5-HT is a neurotransmitter. All of the serotonin neurons in the adult gut are descending myenteric plexus interneurons. Well, what happens if you deplete neuronal serotonin? If you deplete neuronal serotonin, what happens is behavior of the gut certainly changes, but it changes because you find out you've changed the entire enteric nervous system. You have fewer neurons in it. So if you knock out TPH2, which is in serotonergic neurons, you have fewer total neurons in the gut, and particularly this is a marker tyrosine hydroxylase for dopaminergic neurons, you have much fewer of those. So here you have fewer total neurons and many fewer dopaminergic neurons. And if you do a double knockout and produce a mouse that has no serotonin at all, so you take out both forms, the nervous system is reduced about the same as TPH2 knockout, and the same is the effect on the dopaminergic neurons. So all of the effect on neurogenesis is due to neuronal serotonin, not due to the epithelial serotonin, although, as I'll say later, that has an odd effect. Now, with an importance of serotonin, it has to be regulated within the bowel. And serotonin turns on a switch, so something has to turn the switch off and remove the serotonin. And that depends on the serotonin, SER, reuptake transporter, SERT. And that serotonin inactivation is SERT-dependent. Well, what is SERT? SERT is a pump that's present in the plasma membrane of serotonin neurons and other kinds of cells, such as epithelial cells and platelets. So serotonin is synthesized from tryptophan, which is connected by tryptophan hydroxylase, 1 or 2, to 5-HTP, 5-hydroxytryptophan. Aromatic amino acid decarboxylase converts that rapidly to serotonin. A second pump, VMAT2, puts serotonin into vesicles. If it doesn't go in vesicles, it's rapidly inactivated by contact monoamine oxidase, which is on the outer membrane of mitochondria. Vesicles release serotonin. It acts one of 7 classes of receptor. Pump then goes SERT, then into pump serotonin back in. And then once it is retrieved, it can either be put back into vesicles and reutilized or metabolized. So increasing SERT activity, the pump that inactivates serotonin, will decrease the effects of serotonin after it's released. And deletion of SERT or changing its activity will increase effects of 5-HT. So in essence, if SERT goes up, 5-HT goes down. If SERT goes down, the effect of 5-HT goes up. That's easily understood. So what happens, what is the importance of SERT? If you put SERT and look at SERT knockout mice, which don't have any, you find that there are more neurons in the gut than in control, more dopaminergic neurons, and that's true both in the submucosal plexus and in the myenteric plexus down below. It's also true for GABA neurons, and these neurons become a greater proportion of the total. So late-born neurons, this is CGRP, late-born neurons that arise after serotonin in the sequence of development of the gut are serotonin dependent. That is, their birth is influenced by serotonin. Now, you can knock out SERT individually, either in the mucosa or in the myenteric plexus, that is in the CREST-derived cells. So this is a strain of mouse that we created in which we're using CRE-LOX technology to delete SERT only in the enteric nervous system. So SERT is produced and is present in a large amount in the mucosa as well as in the enteric nervous system. And in these animals, this neural CREST driver takes it out only in the enteric nervous system. When that's done, you find that there are many more neurons, but curiously, there's a tremendous effect on serotonin neurons themselves. So usually, here's a wild type, there's a serotonin neuron, and here's the merge. And you see, you can look at a ganglion, maybe you'll get one neuron in the ganglion. And you can see SERT is active, and this is a normal mouse. In the knockout mouse, you see there are more neurons than in the control. And here in this ganglion, there's 1, 2, 3, 4, 5, 6, 7, 8 serotonin neurons. And of course, because we've knocked out SERT, no SERT. So it looks like, and this is very curious, that serotonin neurons stimulate their own development, or, which is more likely, there's another source early in life of serotonin before serotonin neurons are even born that stimulates their birth. Slide are a little whack because I moved them from a different computer. Okay, so neurons are this type of mouse, yet another mouse is here. This mouse has a mutation within SERT. So alanine is substituted for glycine at position 56. And we call these super SERT animals, not because they're super strong, but because SERT is super active. So if SERT is super active, then what that means is that there's too little serotonin. Remember, to take out SERT, you get more serotonin, you get more neurons. This is super SERT, less serotonin. Do you get less? And the answer is you do, and it's massive. Now, you see that here, this is wild type in blue, and the mutant animals red, and that's present for total neurons, dopaminergic neurons, CGRP neurons, and in the myenteric plexus, total neurons here, as well as GABA, and becomes a greater percentage. And you can see pictures of them down here. Now, the reason this particular mutation is of interest is that it occurs in humans with autism. It's associated with autism, and it's over-inherited in children with autism. About 2%, of all children have that particular mutation. So this is a human mutation associated with autistic spectrum disease put into mice. And when that happens, you get mice that have, as I'm showing you here, hypoplasia of the enteric nervous system, too few neurons. Now, interestingly, because these are mice, you can ask them, can you overcome this? Well, the reason that you would have too little serotonin if SERT is overly active is that the SERT is removing it too fast. Well, suppose you took a drug, and we've tried this using prucalopride. Prucalopride is a drug that is available for treatment of irritable bowel syndrome with constipation. It activates the 5-HT4 receptor. Prucalopride can activate that receptor, but in contrast to serotonin, it's not taken up by SERT. That means prucalopride might be able to substitute for serotonin, and if SERT is overly active, it won't matter because prucalopride can't be removed. And it turns out that it stimulates enteric neurogenesis when you try it on enteric neural CREST-derived precursors. So here's a control. These are total neurons growing in culture. Add prucalopride, you get more neurons. You put in a D4 antagonist, this GR compound, reduces it back to the control level. This is the effect of serotonin. It's about the same as prucalopride. 5-HT4 antagonist blocks the effect of serotonin, has no effect of itself. Even greater effect on dopamine neurons. Here's the control. Greater to prucalopride, blocked by the antagonist, serotonin, also blocked by 5-HT4 antagonist. Same effect on GABA neurons, and it's just selective to this group of neurons. Another neuron, calretinin-containing neuron, is totally independent of serotonin. So that means dopaminergic neurons, GABA neurons, and not only under serotonin control, but the critical receptor is prucalopride, and that's not a substrate for SERT. So we tested the idea that if we gave prucalopride during development, we could rescue these mice. And the interesting thing is that it would make you wonder whether prucalopride, given early enough to children on the autistic spectrum, could it, if it didn't cure these, would at least help their bowel. So administered to dams, that is pregnant mice, from gestation through the time of weaning, and the mice were examined afterward. And this is a complex slide, but what it tells you is that the experiment was a total success. Here's wild type. Number of neurons is reduced in these mutant animals. You give them prucalopride, it comes back to normal in the submucosal plexus. The dopaminergic neurons are retained. Late developing CGRP come back. If you look at the myenteric plexus, the same thing happens GABA. And you look at the behavior of the bowel, total GI transit is very much impaired because there are two neurons in control mutant mice. They have slow transit constipation, and you can fix it with prucalopride. The rectal ejection colonic motility is very much delayed with prucalopride. And if you look at the peristaltic waves in the bowel in vitro, you can see that frequency, velocity, and propagation, the ability of the waves to go down the gut, are all impaired by that mutation and all cured by prucalopride. One critical factor then of SERT is to regulate serotonin. If it becomes underactive, you get too many neurons. If it becomes overactive, too few neurons. The critical receptor is 5-HT4, and the overactivity of SERT accompanies at least some children on the autistic spectrum. Well, SERT, as I mentioned, is also present not only in the enteric nervous system, but in the mucosa. And you can see the expression of SERT in the epithelium here. This is a mouse, again, using CRE-LOX technology. We have knocked out SERT only in mucosa, and that's done by using VILLIN-CRE. So they have no SERT in the mucosa, but they have it in the ENS. And so looking at the wild type, you see the numbers of animals here, and shockingly, overactive neurons, more neurons, even though the SERT is knocked out only in the mucosa. So what that shows, and that's true of serotonin neurons particularly. So what that shows is that the role of SERT in the mucosa is to keep all the serotonin that's produced by EC cells confined to that layer of the bowel. It compartmentalizes it. If you knock out mucosal SERT, the SERT overwhelms the nervous system and flows and stimulates neurogenesis. And so the effect of knocking out SERT in the mucosa is identical to that of knocking it out in the enteric nervous system itself. Okay, let me just change position now and talk for a moment about oxytocin. Oxytocin has been called the love hormone because it's involved in childbirth and breastfeeding. It's also a CNS transmitter, which is associated with empathy, trust, and relationship building. Levels of oxytocin increase during hugging and orgasm. It's also produced, we've discovered, within the enteric nervous system. So it can protect mice from colitis. It can protect newborn mice from enterocolitis, but it also has an effect on neurogenesis in the gut. So here's enteric oxytocin. It's present in myenteric neurons. You can see it being synthesized in the gut here. And the oxytocin receptor, there's only a single oxytocin receptor. It's expressed in enteric neurons and in the epithelium and muscle in the gut. You can see it expressed in the epithelium, particularly at the junction of the crypt and villus, and there's a lot of it within crypts. And it's expressed highly during development, particularly in CREST cells as they come into the gut, both oxytocin and the receptor. And the level of it goes down later in life to about 70%. And here's an oxytocinergic neuron. But you see, although there are only 2 to 3% of neurons in the gut contain oxytocin, they synapse broadly within the gut. And you see these little red dots. Each of those is an oxytocin synapse. So you can take out oxytocin receptor in oxytocin knockout mice, and the effects of knocking it out in the epithelium alone with a global knockout or just in the enteric nervous system are the same. So here's the expression of the oxytocin receptor. These are total neurons, and this is the knockout. So oxytocin signaling slows GI transit and decreases stool output. So if you give oxytocin, if you look at the oxytocin receptor here, look at GI transit time, you see it's faster in animals that don't have it. So it's faster in knockout than in wild-type mice. If you give exogenous oxytocin here, you can slow transit time, but its effect is lost if you knock out the receptor. Curiously, enteric oxytocin signaling limits enteric neurogenesis and inhibits peristaltic waves. So the ENS becomes hyperplastic, too many neurons, if you have lack oxytocin. So an oxytocin inhibits mucosal activation of neurons, and that effect is mediated by an effect of oxytocin inhibiting the release of serotonin. It's enhanced if you knock out the receptor and has no effect in knockout mice. So oxytocin is here. You see that it inhibits the frequency of peristaltic reflexes and slows them down. As you see, the ENS is hyperplastic in mice that lack oxytocin only in the receptor, oxytocin receptor. So here's the number of neurons. This is the myenteric plexus, submucosal plexus. You take oxytocin receptor out, only in the epithelium, you get more neurons. You also change how neurons differentiate. How do you get that? That is, how is it that taking out oxytocin in the receptor gives you more neurons? And the answer is that if you take oxytocin out in oxytocin receptor out only in the epithelium, you wind up without SERT in the epithelium. So that's shown here. And so you stimulate the amount of serotonin coming out. You also change the proliferation and turnover of the epithelium. Oxytocin is an extremely important player in the gut. It's present in neurons, and its receptors are present on other neurons and on the epithelium, and it controls the total number of neurons by acting through serotonin. So to summarize what I've said is that the activity of early-born serotonin and cholinergic neurons promotes the development of sets of late-born neurons. Mucosal SERT prevents the large mucosal store of serotonin from overflowing and swamping the developing enteric nervous system. If you selectively delete SERT in either the ENS or the mucosa, you wind up with hyperplasia, too many neurons in the enteric nervous system. And overactivity of SERT, which is associated with the autistic spectrum disorder, gives you ENS hypoplasia. So you could speculate that at least in some children who have constipation and on the autistic spectrum disorder, there might be ENS hypoplasia and slow transit. Enteric oxytocinergic neurons, which also may be important in the autistic spectrum disorder, oppose serotonergic overstimulation of enteric neurogenesis. They do that in part by regulating the expression of SERT. So if you get no SERT, then you get too much serotonin coming from the EC cells. Enteric neurosystem hyperplasia occurs when mucosal oxytocin receptors, therefore, are lacking. So in conclusions, Perturbations of any kind arising in early development, stress, or, for example, that affects serotonergic or oxytocinergic neural signaling, or genes that affect them, can exert lifelong effects on the enteric nervous system and GI function. I put in IBS as a possibility, but any disorder of brain-gut communication can be affected because of the prominent role that serotonin plays in coupling the brain and the gut. And I should also throw in, and the enteric microbiome. Thank you.